All right. Exam on Friday. Any questions about it?
Do you know off the top of your head, like, how many life cycles we've covered that we on this?
There are 9, 10. How many of those practice quizzes did I put up there? Right, so how many, there's your answer. Except we also had the 2 acanthocephalans, Plagiorhynchus and
It was, and then paradoxes.
Paradoxes, yeah, paradoxes.
And then there were no life cycle curves by hand, so that was the one that there were no diagrams for, right? Or there was one that was
Trichomonas.
Like one of them.
Yeah, Trichomonas did not have any diagram just because it's a direct life cycle. Just, and it does, there's nothing really interesting about it. It's, you know, with you put it in a dish and it could replicate with nutrients. Yep.
When will the quizzes be?
They should be available now. So they should have disappeared at 8 o'clock today. And once they disappeared, then you should be able to access all the questions and answers if you took it. If you actually took them. I did find that out. So if you did not take a quiz, then you have no grade, so you can't really click on it. So I'll have to go in and manually add zeros. Same format, right? First 10 will be multiple answer. The next 20 are multiple choice questions. Then we have disease names, matching it with parasite names. Again, you have a word list. Then we've got the diagnosis section, so associating symptoms with a specific parasite, and then also writing the cause of the parasite. So just kind of as a reminder, when we have the cause, it's not, you don't exhibit those symptoms because you have that disease. We need more specificity to it. So we talked about, let's say, plasmodium, and if I said we had patient suffering from severe anemia, the cause is that red blood cells are lysed. They've been lysed. They keep getting lysed. Probably also say it's the lysis and inhibition of new red blood cell production. That could work as well, but we need the cause. And then we have our life cycles, which are going to be tough. But I have all those life cycle quizzes that you could take. Those are going to be good for the practical as well. So again, on the practical, I could ask, what host? What's the definitive host for the parasite? What's an intermediate host? What's vector? So forth. So check those out. And I'm not recording grades on those life cycles. And I think I have it set up. Has anyone tried to take one of those yet? At the end, does it tell you like all of the questions and tell you how many points you got on each? But it doesn't give you the answer, right? No. So then you can kind of go back. The idea is that you can then go back to your notes to find the correct answer. So I didn't want to tell you what the correct answer is. I want you to go back, do the form of active learning for that. So you should be able to do it as many times as you want. I did set, I think, there's time limits on there, and it's based on how many questions. So I think you have a minute for each question, I think is the average. So that kind of makes it a little bit harder. It's meant for you to kind of do it over and over again, kind of as a drill. All right. Other questions? Practicals on Monday. Come at your assigned time, 2 o'clock or 3 o'clock. That's all we're doing on Monday. That's all we're doing on Monday. Yep.
The exam is a campus sample or a plasma?
Yep. Acanthocephala through all of our protozoans. Yep. Already made it. Got it done. Should be printed. And then after the exam on Friday, then hang out for an hour and then go check out the undergraduate research symposium at noon. I'm sure some of our classmates here are presenting. Is anybody presenting? See? Seen that?
They want us to set up at 10.
Tough.
Okay.
Actually, you could probably get there early.
Okay.
Or go right after. So again, on Friday, we don't have a class up here, so I'll probably get up here about 9:45 or 10 till 10. I don't know. Let me check. I can't remember if there's a meeting at 9 o'clock. Let's see here. Check my calendar. No. That's good. Didn't know that. Cool. Yeah, so 9:15 or so, sorry, 9:45, 9:50, you can come in and take it. It's about the same length. So, again, it is the same length. The variation in length oftentimes comes down to how many species we have, how many species we discuss, because that's the disease names and species. So that could fluctuate. I think you've seen it. I think we had, I don't know, 7 or 8 on the last exam. And then also like the diagnosis, I try to go for about 5, but it could be one more or one less depending on how many species we have. Then the final exam is going to take on similar structure. Final exam will be comprehensive, along with some of this new stuff that we're going to talk about today and Monday, possibly Wednesday, we'll see. Plan is to finish up everything on or by Wednesday, so that'll give us like Friday as a question and answer session. So could be a fast course. Ready? Right.
How many life cycles do you want the final again? Like, is it gonna be 2 just like our course, or is there gonna be more?
There would be more.
Okay.
I would probably suggest don't like picking 2 and hope you're gonna get lucky.
No, I have a weird way to set the life cycles. I was wondering how many there will be for us to diagram on the life cycle if you knew already.
On the final?
Yeah, on the final.
I don't know. Best thing I could tell you is we do like generalized life cycles, kind of get those down pat. So even if you don't really know the full life cycle, if you keep the same names in the order that they should appear, you can at least get partial credit for that. All right, and our final exam's on that Monday, 10:30, I think. Get it done, get it out of the way. All right. We are moving away from PowerPoint to something new. So we talked about our parasites. Of course, we could talk about some other parasites. We could go into pentastomes, which we don't really talk about. Those are kind of cool. Kind of an ancient lineage that is associated with reptiles. We could talk about some of those myxozoans, which are that we did hear about, Myxozoa cerebralis, which is the whirling disease. We could talk about all of those. We could talk about some of the microsporidians. We haven't even talked about like arthropod parasites, so the crustaceans or the barnacles and so forth. So there's more that we can do. Instead of moving on to those different species, what we're going to talk about is more parasite ecology and give you terminology. Some terminology and kind of get to some of the mathematical descriptions of transmission. So I'm not going to ask you to actually do the math. I'm not going to ask you to memorize the math. But we're going to kind of present the math and then relate what the various terms mean, because then you can start hopefully relating all of these adaptations that we talked about and said, okay, that adaptation, this is how it impacts transmission. That's what the parasite was adapting to. All right, so kind of start with this idea of epidemiology, all right? Because that is kind of what a lot of people think of when we start talking about describing parasites in populations and parasites in humans. But epidemiology has a very specific term. What is it? Or specific definition. What is it? Zoonotic disease is probably defined it, right? No one's in it. We meet at the same time, right? So I'll give it to you. All right. So epidemiology is the study of disease dynamics in the human population. And that's the term that we know it, or the definition that we know of it as epidemiology. Right or wrong? I think epidemiology takes on a slightly different definition, and it's one that's more in line with ecological epidemiology. And ecological epidemiology is a study of parasite and disease dynamics in host populations. So notice the difference between these two. So epidemiology, when it's just said epidemiology, everyone thinks it's all about diseases in the human population. And we can step back and say, well, okay, well, what constitutes a disease? And conventional wisdom would say bacteria and viruses. But the broader term is really just like anything that causes disease. That would include parasites, it would be viruses, etc. And I don't think we should necessarily restrict it to the human population because these things also go in the animal population. So you go and try to go to a grad program and you can epidemiology, this is what you get. When in reality, it's probably closer to this ecological epidemiology, where it's a study of disease and parasite dynamics in any host population. Could be human, could be wild animal. This term recognizes that we have this parasite dynamics. That's going to influence our host dynamics. So things like increasing pathology, increasing mortality rates. Whatever happens in the parasites, that's going to impact our host, our population dynamics. And then, in the same way, as our host dynamics change, they're going to have an effect on our parasite dynamics. So what this ecological epidemiology, or just like the ecology aspect of it, it's always asking about these things, these arrows. What's causing them? What are the factors that are influencing these areas, the arrows, not just the direction, but also the magnitude of influence. All right, so to kind of get at some of these ideas, parasite ecologists have come up with some definitions to try to standardize our way of talking. And why is that? Why is that? That's because parasites are a little bit different than free-living organisms. Why? Why are parasites different from free-living organisms? If you think about it. Why are they different? What's that?
They don't require a host.
Yeah, free-living organisms don't require a host. How else are they different?
Their life cycles aren't usually very complex.
Yeah, normally free-living life cycles, not usually complex. We do have exceptions, right? Things that undergo metamorphosis. We've got different life cycle stages, but different life cycle stages. That's kind of the rule in parasites, right? You've got different larval stage names, different developmental stage names, and so forth. So we're going to start to kind of define various terms in for the parasite realm. We're going to start with population. So what's the definition of a population?
Number of individuals of one species in an area.
All right. Very good. You must have remembered that from another class.
Ecology.
See? Group of conspecifics occupying a given space. At a given time. You go to that next step with our definition is that it can consist of a single gene pool. So group of conspecifics, just members of the same species. They're going to exist in a given space at a given time. So our definition of a population requires all of our individuals of the same species must be present so they can interact. If they can interact, then they generally occupy the same gene pool. So you're going to have random mating across all of that. All right, this usually works great for some of our host species because based on this, then our life history characteristics will apply to all individuals. In the population. So we haven't defined life histories or anything, but those are things like population growth rates, which are determined by our death rates, our birth rates. We're going to have life history characteristics like how many offspring do we have when we mate? How many times do we try to mate in our lifetime? Do we provide parental care? What is the size of our offspring? And so forth. So all of these life history characteristics should apply to all individuals in our population. We can take the average values and it all applies. Parasites are problematic. So the problem with parasites is twofold. And we already mentioned. So for most of our parasites, we have drastically different life cycle forms. We have adults, we have Miracidia, we have Redia, we have J1s, J2s, J3s, acanthors, right? We can talk about merozoites versus gametocytes, etc. So the parasites have drastically different forms, and we have the other big one. What defines our space? Time is pretty easy. You just go out there and say, at this time period when we're collecting, that's our time. But what's the space? For free living organism, we go out to a pond and sample fish. Our space is that pond. What if we go out to a parasite? Does this necessarily space that pond qualify? Does that term qualify? Maybe, maybe not. We have to account for all these different life cycle forms. So because of this, our parasite ecologists have come up with various definitions and wrote it up in a paper. And got it published as a way to try to standardize all all of our terminology. Like when we say a population, what do we mean by it? When we talk about a community, what's the meaning of it? All right? So, Bush et al. Starts and recognizes a couple different things. We're going to use a complex life cycle, just a trematode life cycle. We go out to a pond. And we sample things. All right. We have our frog. That's going to be our definitive host. We have our snails. That's our first intermediate host. And then we have our dragonfly population, which is our second intermediate host. Now, what we represent is that these big squares represent the population of our host. So this big square is our frog population. Maybe we have 50 frogs out there. That represents a frog population. This square represents our snail population. We probably have thousands of snails out there. And then our dragonfly populations probably in the hundreds. Each of our individuals, our frog population, has the ability to be infected with a different number of parasites. What I'm doing is numbering our parasites. Right, so we've got our generalized life cycle. All right, so our frog, this one right here, this frog has 4 parasites. Yeah, okay. Nope. Our numbers are going to represent different individuals of the same species. All right, that's going to be our key here because we're going to deal with populations first. So this parasite has 4 parasites or worms. This is the infra population. So 4 worms in this individual constitutes the infra population. 3 worms for this individual constitutes the infra population. 2 worms is the infra population for that individual. For our snails, we've got 4 parasite individuals. That's our infra population for that individual. And our dragonfly, this one has 5 individuals. That's our infra population. So our infra population is basically all individuals of the same species that exist in a single host population. But our infra population, the infra component, is representing a single life history stage. So all of these represent our adults. All of these represent First larval stage, all of these represent a second life history stage. So infra population, very specific, it's the same life cycle stage of that same species. And I'll get that, I'll actually write that definition down here. So we're not done with this diagram yet. So, infra population. All conspecifics of a specific life cycle stage in one host individual. All conspecifics of a specific life cycle stage in one host individual. Dragonflies, got 5 dragonflies here. They differ in the number of worms inside, they differ in the number constituting the infra population. But collectively, we can look at what's called the component population to represent all of our conspecifics of a specific life cycle stage in all of the hosts that serve that same role. So an easier way could say that our component population is basically all of the infra populations in a host. Or it's really talking about looking at that one host individual and the dragonflies, and we're interested, hey, what's the population of parasites of that one specific species in our dragonfly host? So note, instead of infra population, we now jump up to a component level. So I'll write the simpler one. All infra populations of a species in an ecosystem, this would be basically our textbook definition. All infra populations of species in an ecosystem, and then because infra populations have a very specific meaning as to what host and what life cycle stage we're talking about, probably a slightly better, more descriptive population that avoids infra populations is saying all conspecifics of a specific life cycle stage within a host population in an ecosystem. So if you remember ecology, Kellen, this component population is somewhat similar to a metapopulation in terms of its structures. Not anything close to its dynamics, but in a metapopulation you have individual subpopulations that exist in distinct patches, distinct habitats. Well, if we can think about our individual hosts as patches, then you have these subpopulations existing within each of these different patches. Now, metapopulations require movement between the individuals, and that doesn't happen between these hosts within that same host population, but at least the structure is the same. Our parasites exist in very distinct patches, and those patches are the hosts themselves. The last term that we haven't described is the entire pond and that term is a superpopulation. So superpopulation is basically all of our infrapopulations of a single species in across all hosts in that ecosystem. Kind of the simplest way to say it. Or you can just say all parasites of a single species in all life cycle and all stages of a life cycle within all hosts of the ecosystem. So that our superpopulation now is all of our parasite life cycle stages, all of our individuals in the various stages in all of our hosts. So now all of these numbers with it constitutes our superpopulation. So infrapopulation, it's the numbers within an individual host. Each host has its own infrapopulation. Within a host species, we have our component population. Dragonflies have their component population, frogs have their own component population. The superpopulation is at a specific habitat. Now we're dealing with all of these infrapopulations in all of our hosts in that habitat. We're now starting to account for all of the different life cycle stages. So we can look at, I think the trematodes probably presents the best terminology. Protozoans are their own thing because they replicate inside the hosts and the various stages inside the host. But we could do it. We could talk about merozoites versus gametocytes. So if we talk about all the merozoites within a host, that's our infrapopulation. If we said all gametocytes within a host, that's an infrapopulation. If we say all of the merozoites that are replicating inside all of us, that would be our component population. All right? And then if we start looking at the vectors, if that life cycle has vectors, and look at those life cycle stages, now we're dealing with the superpopulation. All right? We good with that? These terms also extend to communities. So CD. So our infra community is all infrapopulations in a host individual. So if we change it, instead of saying the numbers represent just different individuals, we could say these numbers represent a different parasite species or a different life cycle stage. So for the honors students, you've looked at, you're looking through snails. When you find snails that are shedding, you're going to find Rhedia inside of them. The Rhedia is one parasite life cycle stage. Maybe you find a Metacercaria. It doesn't matter if it's the same species as that Cercaria. It's a different life cycle stage. Now we're dealing with an infra community. But that only applies to a single host individual. A component community is all infra populations in a host population. So this honors project is really looking at the component community inside of our snails. And we have a component community for each of our species of snails that we're examining. We don't have an equivalent for that supra. Instead, what we do is talk about a compound community. Populations. I'm going to leave it at that. That's all the infra populations and all hosts in our ecosystem. There is, some people would take it one step, kind of narrow this down just a little bit and say we're dealing with a component community within specific hosts, maybe a host within certain life cycle. Say this is a life cycle component community, only refers to these. Others, like myself, says, well, I'm sorry, not component community. Compound community is basically a community out in the pond. Still others, the compound community could say, we're only going to look at the snails and say all of the snail species that are out there, each species of snail has its own component community. The compound community would be all of those infra populations just in our snail community and exclude your dragonflies and your frogs and so forth. We're going to leave it just at compound community because that's normally what we stop at. So we can say we're dealing with the component community. Compound community might also apply. So populations, community, generic terms still apply, but we have to account for the fact that our parasites have different life cycle stages and they infect different hosts, and those hosts are those patches. Questions? Now, because they're in the patches, we've basically said all of the individuals here are infected. Are there ways to describe the pattern of parasites in our host? And yes, there is. All right, so we have statistical descriptors. In some cases, they're very similar to what you would see in the epidemiological literature. Other cases, I'm going to say that epidemiological literature bastardizes a lot of our definitions, adds a lot of confusion to it. They think they're being super clear, but again, they're only focusing on human populations, and that's it. So prevalence describes the percentage of hosts that are infected. So it's the percentage of hosts that are infected by a parasite. Usually, our prevalence term it is given for a specific life cycle stage in a specific host population. And I do try to maintain that. I do try to maintain that. So when we talk about prevalence of infection in snails, we're normally talking about prevalence for a specific type of cercaria. I'll use the metacercaria since those are pretty common. We have echinostome metacercaria. You would report a prevalence for that echinostome metacercaria. That's a specific life cycle stage. It's in a specific host population. We're reporting it for FISA or we're reporting it for Elosoma. If we were just looking at overall number of snails that have metacercaria, sometimes you'll see prevalence used interchangeably with that, but it's probably best to say overall infection is X%. So 90% of our snails have some metacercaria of prevalence. So prevalence is a percentage of hosts that are infected. Pretty easy to calculate. It's just the number that have a parasite divided by the total number of individuals that we looked at. Very easy. So number infected over total examined, and normally we multiply by 100 to get to a percentage. You don't have to get to a percentage if you don't want it. Mean abundance is another term. Mean abundance is the average number of parasites of a specific life cycle stage of a specific life cycle stage in the host population. It does not account for a probability of being infected. It's just simply the average number of parasites. So our mean abundance is total parasite stages found, so total number of parasite stages, specific stages, over number of hosts examined. All right. This means that when we do this, our zeros are included. Our zeros are included in the count. I don't like that, because we can recode a lot of that thing to zeros and ones, in which case, when you take zeros and ones, that value now becomes our prevalence. And in a lot of cases, when we talk about our transmission, it's actually a two-step process. The first step is that our host has to be able to be infected in the first place. So you just have this probability of being infected. And you can say, well, what determines that? Well, it's location. If you don't ever encounter your parasite, you're not going to be infected. So you're just like in completely two different ponds. Or you could be immune, could have already had it, exhibit immunity or premonition or concomitant immunity, etc. Right? Any of that stuff. So you have that first step. Can I be infected? Yes or no. And then if you can be infected, now you have this possibility of, am I infected? And if so, by how much? There's a lot of more like statistical complexity that goes into it. Mean abundance is just your overall average, and zeros are included, and that's probably not the best. I would argue it is not the best way to report it. And instead, what we really want is mean intensity. All right? Mean intensity is the average number of parasites of a specific life cycle stage in, we'll say 4 infected hosts. So now what we do it's the same numerator. It's the total number of parasite stages over total number of hosts that are infected. So instead of total number of hosts examined, we're only dividing by the total number of infected. So now we can report two different values. We can report a prevalence, which tells us what proportion of our hosts are infected, and then when you are infected, what's the average number that you have that's our mean intensity. These are specific terms. You'll also hear a term called worm burden or parasite burden. And normally, parasite burden could take on either of these meanings, all right? The person has to define it. Are we talking about all of the hosts, or are we only talking about the infected hosts? And the worm burdens normally exclude the specific species. So overall worm burden could be all of the acanthocephalans and the nematodes and the flatworms and everything that lives inside of that host. It just kind of depends on how the researcher defines it. Questions? This could be useful, right, for some of you. All right, so we're going to wrap it up here. We're going to talk about some of the population dynamics and get into this mathematical description of transmission. We're going to give it to you, and we're going to try to relate that math, that equation, to the various adaptations. For those of you in honors, if you're wondering, I'm running out there probably in about 30 minutes to go get snails. Tomorrow should be the first day that you can get in there to start looking at them. You need to give them at least 12 hours, preferably about 24 hours. Actually, it's just that morning. Sometimes they respond to light, so time of day, but we'll have our next batch. And please study.